Hello guys, my name is Muhammad Hadi Ali. I'm a 6th year med student at Beirut Arab University and I'm going to cover some topics about inflammation with you. Today's topic is the phagocytic mechanism of white blood cells. I hope you'll enjoy it, and if you have any questions, don't hesitate to email me at my email over here. So first, what is phagocytosis? Phagocytosis is a fancy word for when certain special living cells called phagocytes literally eat other cells or particles. Think of them as the Pac-Man of your body. It eats up anything harmful. We have two main phagocytic cells, which are the macrophages and the neutrophils, that engulf and degrade foreign particles like bacteria, then present antigens to initiate an adaptive immune response, especially the macrophages. So why does phagocytosis matter? Phagocytosis matters because it's a central process to the innate immunity. It's crucial for clearing infections and removing the debris, and step by step it's bridging the gap to adaptive immunity, making it vital for understanding disease pathogenesis and immune system function. And many students make the mistake of thinking that neutrophils and macrophages have the same roles. Well, they have equally important roles, but they have different roles. Neutrophils are like the quick emergency responders, while macrophages are more like the long-term cleanup and communication specialists. Now here we'll see the phagocytosis step by step. First, we have chemotaxis and adherence of the microbe to the phagocyte. As you see here, the microbe, this brown particle over here, is coming, or actually the phagocyte is coming next to the microbe step by step, and then it would ingest the microbe. After that, there's going to be a formation of something called phagosome. Then the phagosome will fuse with a lysosome to form a phagolysosome. Step 5 is the digestion of ingested microbes by the enzymes. And step 6 is formation of residual body containing indigestible material. And finally, there would be the discharge of waste materials. Our first step is chemotaxis and activation. Imagine there's an injury or infection. Damaged cells or pathogens release chemical signals like a scent. White blood cells, especially macrophages and neutrophils, are drawn to the site of infection or inflammation through positive chemotaxis, which is, like we said, you could consider it like a scent being released by the damaged area. So here the macrophages and the neutrophils would be following chemical signals released by damaged cells or pathogens. Upon arrival and recognition of foreign particles like bacteria, the phagocytes become activated and are ready to engulf these microbes. The positive chemotaxis, as a definition, would be the directed movement of cells, which are macrophages and neutrophils in this example, towards a higher concentration of a chemical signal, as we said, guiding them to the site of infection. Next is engulfment. After the phagocyte was activated with positive chemotaxis, the phagocyte starts to extend temporary arms or feet called pseudopods. These pseudopods would surround the foreign particle and then it would fuse after surrounding it. These pseudopods would surround the foreign particle and then it would fuse after surrounding it, internalizing the particle within a membrane-bound vesicle called a phagosome. So pseudopods are temporary projections of eukaryotic cells used for movement and engulfing particles during phagocytosis. The phagosome is the vesicle formed around the particle engulfed by a phagocyte, which is the result of the pseudopod fusing with each other. The third step is phagolysosome formation. Once the phagosome is inside, it quickly merges with another organelle inside the cell called a lysosome. Lysosomes are like tiny recycling centers filled with powerful digestive enzymes. When the phagosome and lysosome fuse, they form something called a phagolysosome, which becomes a superacidic compartment where the breakdown of the engulfed particle will happen. As you can see here, the lysosomes are the organelles within the cells that contain hydrolytic enzymes capable of breaking down waste materials and cellular debris. Hydrolytic enzymes are like proteases, lipases, nucleases. They are found in the lysosomes that break down complex molecules into simpler ones using water. And finally, the phagolysosome is the cellular organelle formed by the fusion of a phagosome and a lysosome, where engulfed particles are degraded. Step 4 is bacterial degradation. Inside the phagolysosome we talked about, the hydrolytic enzymes from the lysosome get to work, breaking down and digesting the engulfed bacteria or foreign material. This process effectively wipes out the threat. After the digestion, only tiny fragments of the bacteria remain, especially their antigens. Antigens are like unique molecules that can trigger an immune response. Now for step 5, it is antigen presentation and release. Macrophages are key antigen presenting cells. They present the antigens to the immune system so that the immune system would recognize it further on. After degrading the bacteria, they process the remaining antigens and display them on their cell surface bound to MHC2, which is major histocompatibility complex class II molecules. This presentation is crucial for activating the adaptive immune response. On the other side, we have neutrophils. As we said, neutrophils are like the emergency team. They are primarily focused on rapid killing and typically release the degraded antigens from the cell via exocytosis. They don't present the cells. So we have antigen presentation, which is the role of macrophages, and we have the release of the organisms, which is mainly done by the neutrophils. The MHC2, which we talked about, is a type of protein complex found on the surface of the antigen-presenting cells, which are the macrophages, that displays processed antigens to helper T cells initiating an adaptive immune response. Exocytosis, which was the role of neutrophils, is the process by which cells release substances from inside the cell to the outside by fusing a vesicle with the cell membrane. As you can see over here, we have the digested microbe inside the macrophage, and then the macrophage would present the antigen of this microbe using the MHC class II, and then a helper T cell would come from here and would now get to know this antigen for further immunity. Finally, we have step 6, which is the migration to lymph nodes. Following phagocytosis and antigen processing, both macrophages and neutrophils migrate from the site of infection to nearby lymph nodes. Lymph nodes are small, bean-shaped organs of the immune system that filter lymph and house immune cells where adaptive immune responses are initiated. You could consider them like the headquarters of your immune system. In the lymph nodes, macrophages would present the antigens to specific T lymphocytes, thereby initiating a more targeted and long-lasting adaptive immune response against the pathogen, as we mentioned earlier. The adaptive immune response is a specific immune response that targets particular pathogens and develops memory for future encounters. Let's quickly review the most important points about phagocytosis. As we said, phagocytosis is a foundational or fundamental part of your innate immune system, acting as the first line of cellular defense against invading pathogens, and it is for clearing cellular debris. It is also a critical link to the adaptive immune response through the antigen presentation on the macrophages. Key phagocytes are macrophages and neutrophils, which are the primary phagocytic cells. Each one of them contributes uniquely to this immune response. Neutrophils are for rapid initial killing, and macrophages are for sustained phagocytosis and antigen presentation. And what ensures efficient elimination of the foreign particles is the sequential steps of engulfment, lysosomal fusion, and finally enzymatic degradation. And there you have it, the incredible journey of phagocytosis, where your white blood cells act as tiny guardians, keeping you healthy and safe. Thank you for your time.